Okay, to start, could you each tell me your name and your most recent position?
I'm Bob Waterston. I'm currently a professor of genome sciences at the University of Washington School of Medicine.
And I'm Jane Rogers. I've now retired, but my most recent position was working as a consultant with the International Wheat Genome Sequencing Consortium to deliver a sequence for hexaploid wheat.
Could you each talk some about your respective roles in the Human Genome Project? What are you each most proud of, and what do you consider to be, in hindsight, the most significant roles?
I worked with John Sulston at the Sanger Centre. John was director of the Sanger Centre at the time. And we worked on the building up the sequencing facility so that it was a large-scale facility. And I took over responsibility for delivery of the Sanger contribution to the Human Genome Project, the generation of the data and integration with that was then integrated with the other centres. And the Sanger, looking back at the history, I think played a number of significant roles in the actual project, ranging from helping to develop the strategy for the whole project based on the maps and the clones. And I think we also played a significant role in keeping the project as an international consortium project. We took the lead with WashU, Bob's group at WashU, in making data freely available as soon as possible after the sequence was generated. And we also made a very significant contribution to the finished sequence. And I think I think all of those are really very important roles. For me, the contribution that we made to the finished sequence and generating a very high quality product that has provided an excellent foundation for human biology since then is a major contribution.
Bob? Yeah.
Yeah. Well, as you know, I led the Genome Center at WashU, which, along with the Sanger, represented the two arms of an international consortium. And I think together, as Jane said, we worked to make the project truly international. And, WashU played a leading role in several areas. We'll get into them later, I think. I did everything from lead the center to make the subclone libraries for a long time. I think most of those libraries I had a hand in. Anyway, what am I most proud of? Obviously, our contribution to the sequence and the finished sequence, as Jane said, was really critical. And remarkably, that was the basis for, I think, HG37, or I can't remember the exact acronym ahead of time, but 37 is the version number. And it's still being used. It's the primary source for most people today. It's remarkable, 20 years on, that's still, it's going to be replaced very soon by the telomere to telomere sequences, really finished the whole genome sequence, but it held up very well. But I think the thing I'm most proud of is our successful effort to get the sequence in the public domain quickly. The Bermuda rules came out, I think, from basically the history of the worm. John and I advocated strongly that the sequence had to get out there quickly and without patents. And that was based on our very successful experience with the worm sequence. And I don't think people other groups, certainly the human genetics community, is that kind of thing is very foreign to them. And the Alberts Committee, I think, thought of sharing, but they thought of sharing as sharing materials and sharing things between centers. They were not thinking of this very rapid public release of the information. And I don't think they talked about patenting at all. And with the Bayh-Dole Act and things like that, I think it was very important that the HGP took a stand on that. So I think, again, together with the Sanger, that was something that I'm very proud of and has an impact on other science projects today.
No, I think the whole sort of data release is one of the essential legacies of the Human Genome Project, which really continues to be impactful to this day.
No, I agree. And I think you really can trace it right back to the worm. And we did it because the worm community was a community of sharing. You know, there was the Worm Breeders Gazette that got published, and we put in one-page abstracts of what we were doing, unpublished stuff. There was an agreement, basically, that nobody would take advantage of that. And it was a good community, and we shared. And John, when he started the map, made it an explicitly community linked endeavor. And we continued that with the sequencing.
So could you both give me a sense of the significance of the initial sequence and analysis paper? And do either of you recall your initial impressions about the significance of sort of these efforts in 2000 and 2001?
Well, I mean, there's the paper and there's the sequence. Those are both important items. I mean, and the sequence was really spectacular. I mean, it was a draft sequence. It was crappy. We were missing lots of things. There were holes in it and everything, but it provided a view of the landscape, the whole landscape for the first time. And I can remember looking at sequence coming off the machines and popping it up on my computer and thinking, you know, this is 4 billion years in the making, and I'm the first person to ever look at it. I couldn't make sense of it. It was just A, G, C, and T, but there it was. I was looking at it, and to be reading our genome and have it all there. I mean, well, not all, but almost all there, was spectacular. And the paper gave us was a first pass at trying to understand what's in there. It was, I was awed by it in the sense of 65 pages, and nature took it all. And you know, we'll come to it maybe. You know, there were mistakes and so forth, but it was a serious effort at trying to understand what was there with all its limitations.
But I can remember also being so impressed at how many analyses could be done with the draft sequence. I mean, it really was, you know, quite amazing to come up with what we did. And as Bob says, to have the global view and also to have tools that began to allow you to access different parts of the genome sequence, and to be able to view it at different levels that are on a sort of, you know, whole genome scale down to the individual sequence data.
Yeah, Ensemble was already trying to give us access to the sequence in a convenient way.
Yeah.
Yeah.
Both of those are really, really interesting expositions of the significance, and I think the sense of it as imperfect but still meaningful, and according to a number of registers, is really well backed by by posterity in many ways, and is also reflected in the materials that we have in the archive as well.
But I went back for this interview and looked at the paper, and I looked at the list of major conclusions, and they're not very impressive, actually. I mean, we talked about GC, GC rich islands, and well, that was already known, and we got the number of genes wrong, and and we had bacterial contamination that we called horizontal transfer. but at the time, it was really awesome. And I think it really was not the specifics, but this idea of a global view of really knowing where we are in the world.
Yeah, I think that's a really wonderful way of putting it. Jane, any further thoughts?
No, I think I agree with what Bob said, and I suppose the other thing that really had an impact, was the fact that we were interested. Everybody had an interest in this potentially, because we all have a genome. Well, we've all got multiple genomes, but it's relevant to all of us as human beings, and I think this was the first time that a genome sequence had this much relevance to our curiosity about who we are and where we've come from.
So my next question is around sort of both of your involvement in the actual sort of development and writing of the paper. I suppose we could start with, your thoughts about how the paper took shape, and your sense of sort of who was decided to do what sections, and your reminiscences about sort of the discussion of the sort of division of labor in the writing and the conceptualization of the piece.
Well, for this, I think Bob was far more involved than I was. I was much more involved on the generation of the data side of things, and at the Sanger, the people who were deeply involved in putting the genome together, and certainly doing gene calling and sort of developing the algorithms for that were the bioinformaticians. So Tim Hubbard, Michelle Clamp, Ewan Burney, and Richard were involved in the discussions, as part of the analysis group. But Bob, you're a curator, so
yeah, you are a manuscript curator.
Let's step back from the paper. You asked for how it took shape. There was controversy about what kind of paper there should be. For the worm, we had gotten the sequence out there. We'd done a fairly brief analysis of things, and then we enlisted others to write papers on, and we made the sequence available to them, and they wrote papers on different aspects of things that they could find. And John Sulston was of the view that something like that should be done with the human, and what's more, it was just a draft, and he wanted to get it over with, get it out there quickly. He wanted a September publication, and have it be fairly brief, and then let the community have at it. And that's the nature of, that's the way John viewed these things. And Eric, on the other hand, was like a kid in a candy store. He wanted to get his hands on it. He wanted to see what he could see in it. And so there was some back and forth about what kind of paper it would be. And John eventually conceded he could see that with Solera doing their thing and so forth, we had to be there with a decent job of analysis. We had to know what was in our product, and we had to know the product was good enough to do this kind of stuff. And so with that, Eric took the lead. I think there's some quip about John telling him, if you want to do it that way, Eric, you write it. And I think that's right. I think I actually remember that conversation. And so Eric organized what became known as the analysis working groups. And we had a phone call, I think, every week. And somebody would be tasked to look, the people that Jane mentioned from the Sanger, people from WashU, people from all over the place. Eric invited people, and I think it was, I don't think there was an open call for people, but anybody who thought they could contribute, I think, was welcome. And then they were tasked with presenting a figure of what they wanted to talk about and a paragraph or two about what they had found. And so Arian Smith talked about, dove into the repeat sequences, and people, Ewan was very involved in gene predictions. And, we went through each of these topics, GC, the GC rich regions, and all, each of these topics came up, and they would get discussed. And Eric led the calls. He was a vigorous critic. I think I had a good input on some of those things too. If I couldn't understand it, the reader wasn't going to understand it. And so this went back and forth for months, with things getting refined. They would be presented once, and then they'd get critiqued, and then oh, this figure looks crappy, you should do this. How about this kind of representation of your data instead of a bar graph? And things went on like that. And so, and I don't know, in terms of how the specific tasks, I think I think it comes up maybe in one of your other questions, but we'll deal with it. I can deal with it here. Francis, I think, was tasked with writing the introduction. I think that's right. I was tasked with writing the methods and summarizing just what the sequence was. And Eric took the analysis. And I don't know what John's responsibility was. Do you remember, Jane?
I don't. I think it was probably.
Have you covered what John's responsibility was?
I haven't. Probably overall editing, I would think.
Yeah. I mean, John was a very good writer. Anyway, so things were divvied up that way. And then Eric took all these different vignettes and stitched them together into a paper. And then I edited heavily what Eric wrote, and Francis had to go at it too, and so forth. And then Phil Green reviewed it and told us how we should have done the paper.
I think most reviewers are like that, actually.
No, this was.
Oh no, Phil wrote
40 pages or something.
I think it was 17 single space pages.
Oh, right.
I don't know. It was a very long and detailed critique of and told us, on this analysis, you should have done this statistic instead of that. I mean, it was very detailed.
So that kind of critique. I wonder if he still has a copy of it, because that would be.
I probably do.
Then. Yeah, I actually, that would be a really valuable. I would, if you don't.
You haven't heard of that before?
Not that explicitly.
Well, Phil had deliberately not partaken in the analysis and so forth because I don't know. he likes to work alone, partly. He's not very good. He has a history with Eric. That might have been part of it. I don't know. But so he was an objective reviewer. And we were lucky to have him. He had very good suggestions for us.
Could you just, as Bob's been talking about the manuscript, could you just describe what it was like in general, just working with Bob, John Sulston, Eric Lander, and Francis, and other leaders on the HGP during this time?
The time was not, I think the time that you really are referring to is the time when the project really came together and it accelerated. So this was, stimulated by Solera making their announcement. The HGP agreed on a strategy and how we would go forward. But at that point, the time we had to deliver it was, we had a short time to deliver it. And we had a number of major obstacles. Generating maps for all the chromosomes was one significant hurdle. Scaling up the sequencing was another significant hurdle. Applied Biosystems had come up with new sequencers that would make the job easier, and those were the sequencers that Solera was going to use. But for those of us who were already sequencing, this presented a challenge in having to change over all of our technologies to adapt to the new sequencers. And I have to say this because it was always my beef, and the sequencers, as they came, did not work properly. So there was always the challenge of actually having to get them working efficiently at the same time as working up everything else to match with them. So, I suppose, what would you call it now? You'd call it a perfect storm now of, lots and lots of pressure to get things working, deliver. And the sequence had to be, very efficient. We had targets to meet. We met on phone calls once a week, and we had to report amongst the what we glibly amongst ourselves called the G5. So these are the 5 largest sequencing centres, and we had to report on what our progress was. And what was it like? Well, it was obviously, it was stimulating, it was challenging, but at times we used to have these calls timed for Friday afternoons. And certainly over the summer months in the Sanger on a Friday afternoon, because we're very close to what is now the Imperial War Museum in Duxford, if there were air shows over the weekend, then we would have the sounds of Spitfires, Lancaster bombers, the old World War II planes in the background of these calls. And sometimes the negotiations with our collaborators in the United States, I have to say that the way that commanders during the Second World War must have felt at times when they were sort of planning the advance across Europe, certainly came to mind, because it was a tricky situation at times that we had to handle and make sure that we were all pointing in the same direction and sort of going on the same course. And there were times when it looked as though the course would deviate, and at that point there had to be another reckoning, a lot of meetings, a lot of email exchanges, and then we worked to get back on course and go ahead again. But it was challenging. It was challenging to get it done.
And one issue that comes up pretty frequently is the quality assessments and making sure that the sequence is accurate. And could either of you go through a discussion about how there was all of this quality assessment going on and how that process went, and how you decided essentially what the draft sequence standards were, and how you would present that?
Bob, you talk about this. It was a new metric every meeting.
New metric, that's right. And I certainly, there was game playing to make sure that whatever metric came up was one that favored what you were doing. And some of us are better at that than others. And I don't know, it was important. We had different groups and we were proceeding in different ways. We had to come up with metrics. We had to come up with fair estimates of costs so that we really knew what things were doing. The Sanger was blessedly outside of this, and they were forced to listen to us hash through this stuff among the NHGRI folks in particular, because NHGRI wanted to know if its grantees were performing. And so we had to we had to do these endless reports. I don't know how much more sequence we could have done if we didn't have to do the reports, but but maybe, and so we had to figure out what the base quality, what would count toward your production. And I don't know, it was endless.
I think that one of the final assessments in terms of the quality of the overall sequence that we were producing, we would assess how many contigs each of the backs were assembled into once we put them together. And I think we ideally, you'd aim for, I don't know, maybe up to 10 gaps in a shotgun sequence of a back. That was generally a pretty good sequence. Some didn't go together that well at all, and maybe needed some more. But another metric was the depth of the sequence. I mean, we were aiming at fourfold coverage on the draft sequences. Sometimes, if the clone was small, you generate a bit more. If the clone was big, sometimes it got thinner. And again, ideally, you topped it up and put a bit more in there. But I think we measured all of those criteria, and then when we came to generate an assembly of the clones, so we looked at the overlaps in between clones, and that would also give us an idea of the quality of the sequence, how well those went together. But it was largely I remember, very dependent on the sequence depth. And I know John was very impressed with the idea that we have to count the cost all the time. So we had to generate the same cost measures, and also the NHGRI was very interested in how we measured that. So we had to report as well, Bob.
Oh no, I know.
No, thanks for the detailed assessment. That's really helpful because, in particular, some historians are very interested in how things like quality assessments were done and how the various metrics were agreed upon, and what scientifically and sort of community-wise went into those types of shared scientific standards at this phase in the Human Genome Project.
One of the advantages we had, I should get this in here, I think, is that Phil Green had developed his base calling algorithm, and that produced a quality metric for each base, and that was automatic and objective, and that was enormously useful and valuable in these quality assessments. We could rely on that, and it was used across all the centers.
Yeah.
If we'd all been doing different quality metrics, it would have been a mess. Everybody would have been accused of fiddling the quality measures to boost up their numbers and so forth. I mean, the fact that we had this common and very reliable, very robust metric to use was very good.
Right, and some of those metrics that, like for example, the error rate had been in common conversation for a number of years. So that also, I think, probably helped as well. That some of this was built on for a number of years before this huge scale up finishing project.
Well, that was the value of doing things like the worm and yeast and so forth. So you knew what the answers needed to be.
So one thing we haven't talked about at all are some of the companion papers. So for both of you, what were the more significant papers that were published alongside the initial sequence analysis paper? And again, your thoughts on sort of how those pieces of the Nature issue came together, and your thoughts on their importance.
Well, I suppose that I would say that the other big companion paper that stands out is the Snip Consortium paper.
Sure.
This was reporting 1.4 million SNPs that have been mapped across the human genome, and the data were public. And the Snip Consortium had started up in, I think, the spring of 1999. And this was a consortium of pharmaceutical companies who had agreed that a public resource, single database with these data in there would benefit everybody, and it was pre-competitive. So Michael Morgan and the Wellcome Trust did a good job in getting everybody together and organising the project. And the goal was to identify 300,000 SNPs across the genome. That was what the project aimed at. But by the time that the draft sequence was there, we not only were able to call SNPs on the restriction digest fragments that were the original focus of the project, but we could align that to the clone sequences and call SNPs there and call SNPs in the overlaps. So putting that first map of variation across the human genome, I think that represented a huge achievement. It was an important a companion to have there. I'm not quite sure when it was decided that it would actually be able to, the data were there to come to fruition at the right time, but probably the timing was just about right. The sequences were being generated in parallel with the clone sequences. So
yeah. There was always one of the, at least from Francis's outlines, there was always supposed to be a companion paper on SNPs, and that's fairly early. So the significance was realized immediately. Snip Consortium. Oh, go ahead, Bob.
Well, I was going to say it formed it was the impetus for the HapMap. And it really does start to reveal our evolutionary history. It's valuable for human disease, but it's also starts telling us about where we came from. And it was funny, the consortium, the pharmaceutical companies were in on the decisions about how all this should proceed. It was their money. And they wanted this restriction digest limited set because it didn't depend on us having a draft sequence. That was its strength, but its weakness was that it didn't acknowledge that we were going to have a draft sequence. And so it was very inefficient. We had to do lots of sequence to get a few SNPs. But finally, we were able to convince them as the draft was coming along that we could just do random sequences from this panel of individuals, and we could, every read would reveal a new SNP. And so suddenly we were, when we were able to convince them that was a viable strategy, we were able to discover lots more SNPs very quickly.
So just one follow-up question I've always wondered is who came up with the phrase golden path? Was it Jim or was it somebody else? I know it's not the preferred name, but
that got squelched in the writing. I think it was Jim.
I think it was Jim too.
I think it was.
Yeah.
Because he really did have to choose his way through all these clones.
I think another very early rejected name for it was Yellow Brick Road. So it's
that was probably a bad day name.
there's a very interesting history of what things are named before there's some consensus on what to name.
Yeah.
The hat map being a great example of sim map, other things,
Yeah.
shared inheritance for medicine map, and all these things.
Yeah.
But one question that I wanted to ask both of you was to say that there's been at least to me, there's been always a discussion about, whether in order to have good sequence, you always need good maps. Is even with current technology, is that the case, or more recent technology?
So I should have mentioned that when I was talking about the value of the map, because the map provided a way to get the finished sequence. The map had many fewer gaps in it than the sequence. And so you could look at where you had a clone, you could pick the next clone automatically, or almost automatically. I mean, it was there for you, the information was there. And to get high quality sequence, you need to have the territory refined enough to a small area, so you're not dealing with all the complexities. You can target your efforts to the problems in that clone, and they're different. That's why finishing was so hard to automate, was because every clone had its own problem. You did enough of them, you could recognize common problems, and you could start to devise semi-automated ways of addressing those, and then something new would come up. But the clones let you do that, and until recently, there's been no high-quality genome that wasn't a clone-based, map-based genome. The fly sequence was highly touted for being the first shotgun sequence, but they spent 5 years, I think it was, finishing it on a clone-to-clone basis. They had to go back and build the map. The mouse was the same way. We did a lot of clone-based sequencing to start with, but then we shifted to the whole genome shotgun. And I think it wasn't until 2008 or 9, something like that, when the mouse was finally finished, whereas the shotgun was out right away.
No, so earlier than that, because the mouse was finished when I was still at Sanger. So it must have been 2005, 2006.
Was it? I thought it was
it was earlier than that, but yeah.
And I don't know, then the high quality genome stopped coming.
Yeah.
Because Solexa Illumina took over, and it was just too cheap to do whole genome, and nobody finished. And it's interesting. That's going to change because the long reads are basically a substitute for the back clones. You suddenly have a large segment of DNA that isolates itself from all the other problems in the genome, and you can focus in on that, and you can assemble that. And they've actually, with that kind of stuff, they've been able to do things we couldn't do with the backs. They've actually gotten through the centromeres. Anyway, I think that's right. the clones until now have been essential to high quality genomes of any kind.
The other thing that the clone sequence allows you to do is actually check it as a finished sequence. You can do you can do an in silico digest and compare that with the actual restriction digest that you do as part of the subcloning process, and actually compare the two. So you can check that the sequence is assembled correctly with the clones.
Yeah, and that's a step that we did on all our finished clones.
Yeah, we did too. And you know, it was very nice to produce a high quality product that people could use. So many of the other research communities who have had genomes of the organism of interest sequenced, and I think the cow and the pig and so on. There were shotgun sequences, and a lot of the people in the communities don't realise that when they actually want to look at a biological problem, that the sequence quality may not be good enough to actually allow them just to extricate what they need and not do any further work before they start.
Well, or they are faced with doing that extra work. If they recognize that they have to turn around and do the work that we did on the Human Genome Project. Yeah. There's a lot of deferred costs in unfinished genomes.
Yeah, it's a very interesting discussion when you have different communities that are looking at sequence and have different sort of quality metrics for each, sort of comparative grade sequence or human grade sequence, and how right, and how and for a draft sequence may not be very useful if you want to look at evolutionary questions, but it may be useful for some other questions that another community could ask.
And unfortunately, the effort was really never made to fully automate finishing or automate clone maps and so forth. It was just swept away. And so it's true that if you're working on an organism that only 3 other groups are doing, it's too expensive to do a finished sequence, or it has been too expensive. And it's just economics. You have to you can't get the whole genome. You just have to put the effort into where you need it most.
Yeah, it's always fascinating to look at the sequence finishing process having been described as artisanal and other adjectives. It really is a very different kind of work and very different kind of specialization.
Yeah,
in many ways.
Like I said, the problems are varied, and there's lots of different reasons why sequence can fail and why assemblies can fail. Just lots of different ones. And you think you've seen them all, and then there's something else that comes up.
Jane, my next question is to you first. Is when the initial sequence paper was published, what sense did you have of kind of the work left to be done, and with finishing the sequence, and what were kind of the most pressing sort of issues to be addressed in 2001, going from 2001 to 2003?
So I think you have to, because publication of a paper comes sometime after you've actually said you're going to halt the data collection, because that's you halt it, you then have your data for analysis. It was really at that stage when we finished generating draft sequence that we thought, okay, done that, now we have to get on to the job of generating the sequence for the finished sequence. So by the time the paper was published, we'd already been on to the finishing problems for 6 months. But it's the same, the same feeling of, oh, now we've got to go and sort things out. So the problems that we were facing, first of all, completion of the maps. Bob's group produced the whole genome map, but at Sanger we had individual chromosome maps, and we worked on finishing the maps on the basis of using the sequence. So you use that to go in and probe for more clones to complete the maps as you go along. We had a shotgun sequence for a high proportion of the clones. We hadn't actually stopped our finishing process through the whole of the generation of the draft sequence, much to the annoyance of Eric on more than one occasion on the G5 calls. Sanger was sequencing too deep, he's sequencing more clones. But we had to go back and complete the shotgun sequences. And as part of the scale-up process, we thought we could store all the subclones and go back to those and use them for finishing. So we invested in 3 porta cabins. Does that make sense in American? Yeah, 3 porta cabins essentially of freezer porta cabins, for which we had to generate a clone tracking system, an informatics problem, so that we could then go and find these things all stored at -20. The sort of size of fridge that we were told would normally use for frozen peas or something like that. So we had 3 of these stacked outside of the Sanger Centre. So working out how to do all of that, how to retrieve the clones, then we were off shotgunning again, and then doing the hardcore finishing, and we had a lot to do. And in finishing, you have the problems with the actual technical problems, of dealing with the data, getting the everything done to get your sequence together. But if you have, as we did, several groups of finishers who spent their days in front of computers, we also started to had to deal with problems with people having problems with repetitive strain injury and sort of health type things. So you had to make sure that people had breaks, didn't work too long, and so on. So all of these things sort of just added to what you had to deal with in terms of getting the sequence done. But it was 6 months before the actual publication of the paper, and of course when the paper was actually published, a lot of people thought that was it, that was the sequence. And to then say no, we've got another 2 years of working on the finishing. I think there were a lot of people saying, well, what are you doing? It's done. In the end, I think the proof came in the high quality that has stood the test of time. But keeping people motivated also became something of a challenge sometimes. But Bob, there were have I covered the main issues? I mean, sorting out the maps and then sort of filling the gaps and dealing with the tricky problems, because some of the things that were challenging in the draft sequence were not only repeats within clones, but also then large repetitive elements in the genome that and duplications that were collapsed. So that took a lot of sorting out.
And Evan Eichler is still sorting them out.
Yes.
These Eichler repeats.
That's right.
Yeah.
No, I think you covered it pretty well. I mean, when that was certainly, we knew we had to finish this sequence. There was a lot of concern that having captured as much information as we had, that the drive for finishing would be diminished, and we had to push people up front. And when we agreed to do the draft, that people were committed to doing the finishing. And so, like Jane said, we'd done the draft shotgun. We stored all the subclones so that we could go back and use them. We didn't have to resort to porta-cabins, but we had massive storage and access issues. And so we did all that. I would say the other thing facing us was the mouse.
Yes.
Because Celera had quit doing their shotgun at, I can't remember, 4x or 5x or something like that, because they were trying to augment it with the public data. And they figured they had enough between the two. They had turned all their shotgun capacity to mouse earlier than they had planned. And they were, I think by the time of the publication, they were already selling access to the mouse sequence. I don't know when it was formally available, but it was around that time that they were advertising anyway. They were promising it and recruiting buyers. And I remember colleagues at WashU who worked on mouse saying, "Oh, I feel like a traitor, but we're going to have to buy it." And again, this was a real threat to just this whole ethos of biology, of science, I would say, not just to the genome, because if you can do it here, you could do it elsewhere. And so we agreed that we had to make a significant and rapid stand here. And together, the Sanger and we and the Whitehead forged ahead at breakneck speed. Basically, we turned the capacity over that we had developed in the shotgun and Eric convinced us that on this case, we did have to do the whole genome shotgun, and we went along with that because anyway, I don't even know if there were good. I don't know. I can't remember what the status of the libraries was even, but we weren't going to do another effort, a crash effort, and so we did the whole genome shotgun and did a remarkably good job on it, I think. But we talked at the same meeting and something, and he came up to me afterwards and complimented me on the mouse sequence, because it was, Celera had published, by that time they had published the sequence of one chromosome, because they were clearly not going to make any promises for availability for anything else. So they published this one chromosome and made that data available. And ours was much better than that. It just was. We already had a fair amount of finished sequence for the mouse, or clone-based sequence anyway.
I think we were generating that because we had a map.
Did you?
We made the map because
oh no, you
made comparatively.
That's right. We did that together because we realized you could, that's right, I've forgotten that. You could use the back-end sequences and their homology to human to put them together. Yep. And yeah. So we did have, I'm wrong about the clone map. That's right. But we didn't use it. We did the whole genome, but we had combined stuff, and we did have the back ends and so forth. So that was a big immediate challenge to get that out there, and we did. We got it out in remarkable time. And this colleague at WashU came up to me a few months later, chagrined that he'd spent the money. I warned him that it was going to be, it wasn't going to be good money, or it wasn't going to be money well spent, but he did it anyway.
Yeah, I should clarify that in, I think it was maybe April of 2000 when there was the initial freeze for the draft sequence, the data freeze, that already there was very intense discussions about finishing the sequence. So
Yeah, no, we knew we had to do it. Yeah. Well, there were intense discussions about finishing. It goes all the way back to our crazy proposal in 1994. John and I put forward a proposal in 94 to basically do a draft sequence and follow up with finishing. We were going to do a better draft sequence, but we were going to do it more deeply, so we didn't have to go back and do more shotgun for the individual clones.
yeah, this is finishing the sequence 5 years early, something.
Yeah. And one of the big criticisms that we had, Maynard Olson in particular, said, if you did this, it would never get finished. So the idea of there's this tension between capturing the information quickly and not completely versus more expensively and with more effort. And I don't know, Maynard had this saying, or I don't know, a little story or whatever. He said, he was this is Maynard Olson, came in from chemistry, where things are on a much more solid footing than they are in biology. And his impression of biology was that, when you got into this, it was like quicksand. You just kept getting into more and more complexity and more exceptions. And to him, the genome was rock at the underneath the quicksand. That if you had the genome, you could only sink so far. and so to him, it was important. It was vitally important that rock be solid and not full of fissures and cracking and falling apart and letting you sink further and further. And so he was a very strong advocate of finishing. Yeah.
So just because we're a little over time, and I want to be careful with everybody's time. The final question I want to ask both of you is just your concluding thoughts about the significance of the sequence and the paper for genome biology and genome science in general, looking back.
I think it was the first time that it's not the general public, it's the general scientific community could see the value of having genome information. And I mean, on a sort of minor level, you can say that in an instant, Ph.D. projects in human genetics were transformed. Instead of spending 3 years to clone a gene and possibly sequence, you went to the database and you start your PhD project with the sequence of the gene. You can then start to do the biology. So I think it was transformative, with that sort of example.
I would agree. I mean, the yeast and the worm sequence convinced those communities early on about the value of genomics. But for the wider community, it needed the human genome. What was gratifying and surprising to me, I think, was how rapidly that created demand. The zebrafish community had to have their genome sequence. Richard Gibbs had to do the cow for the Texas farm ranchers, and all of a sudden, the revelations that the power of having a genome sequence was obvious to so many people, and it just created an immense demand, which is fueled the field, and it drove Solexa. It created a market for them, and they could get investors. They could bring their product to the market and have buyers. I don't know, and I think it also let people see the value of doing some things at scale. Biology is a cottage industry for the most part. This data-driven science is sort of foreign to biology before all this, and it became apparent how much more you could do with a data-driven approach. And so I think biology, I don't think there's the dichotomy that there was when the Genome Project started. There was a lot of antagonism toward the Genome Project because it wasn't hypothesis-driven. And today, I think people, I don't know, they've incorporated it. It's part of fabric. And so you do both. You collect data at massive scales, and it's, with Illumina technology, small labs can generate more data than they can analyze very quickly. And so you do both. You're able to collect huge amounts of data to drive your hypotheses. And I think it's been very good for biology. It's changed things. And I think that really did come from the obvious value of the human genome. It just, like Jane said, Ph.D. students could all of a sudden zero in on what they needed to study. And I don't know, and all kinds of other things that, like the HapMap followed, and now we have sequencing of ancient DNA, and it's just that the amazing thing.
Sequencing of individuals to look for COVID susceptibilities.
Yeah. No, the amazing thing to me is how pervasive genomics became so rapidly. I guess I was too schooled by the opposition that we'd faced in the early days, that people would be won over so quickly and change the way things were done. It's been amazing.
The democratization of sequencing technology really has been remarkable. Any kind of lab, many labs produce much more data than they can easily analyze with current sequencing technology.
Oh, and we're doing a lot of single cell analysis, and we throw sequence at it because the other part is more expensive. So we use sequence to extract all the information we can out of the data, and it's so available. It's not as cheap as I want it, but it's really available.